Recent demonstration of in vivo imaging of glycans uses a bioorthogonal chemical reporter strategy of treating cells or organisms with alkyne or azide-tagged monosaccharides. Modified monosaccharides, processed by the glycan biosynthetic machinery, are incorporated into cell surface glycoconjugates. Bioorthogonal alkyne or azide tags then allow covalent conjugation with fluorescent probes for visualization or with affinity probes for enrichment and glycoproteomic analysis. This click chemistry-based method allows for the rapid, non-invasive, and robust labeling of alkyne or azide-tagged glycans in zebrafish embryos and can be readily extended to visualize other classes of glycans in developing organisms.
The main advantage of this technique over existing methods, like lectin or antibody-based glycan detection methods, is that it can be applied to dynamic in vivo imaging. Lectin and antibody-based imaging methods only provide a snapshot of the glycans at a particular time point. They are difficult to implement in the context of dynamic studies. In addition, labeling glycans with lectins or antibodies requires removal of the cells or tissues of interest from their native environment and fixing them before analysis. By contrast, labeling glycans using the bioorthogonal chemical reporter strategy overcomes these limitations and can be directly applied to non-invasive imaging of glycans in living systems such as zebrafish embryos and C. elegans embryos. Using BTT-ES-mediated azide-alkyne cycloaddition, a biocompatible version of click chemistry, robust labeling can be achieved within 1 to 5 minutes.
Demonstrating the procedure will be Dr. Jiang and Feng, postdocs from my laboratory.
Collect and transfer